In this video, I'm going to show you how to transfer bacteria from a culture tube onto an agar plate, also sometimes called a petri dish or just a plate. So let me show you what these look like. Here is our culture tube. This has bacteria growing in it in broth. And here is the agar plate. It has a lid that opens, and inside there is this sort of jelly surface made out of agar that is filled with nutrients, sugars, amino acids, and sources of nutrition for the bacteria. And so the bacteria will grow on the surface or even down into the agar on this plate. You're also going to need a Bunsen burner, a striker to light the Bunsen burner, and an inoculation loop. Let's start by labeling our plate. When you label an agar plate, usually Sharpie is the thing that's going to label it the best. You want to make sure that you're not labeling the lid, because that lid, if your agar plate ever falls down on the ground, the lid can become separated very easily, and you might not be sure which lid goes with which plate if you dropped, say, a stack of plates. And so we always label on the side that has the agar in it. In this case, I'm just going to label with my initials. Okay. Now, let's go ahead and get our flame started. We'll turn on the gas and use the striker to light the flame. And what we're going to do is we're going to use the loop to get a loop of bacteria out of the culture tube, the broth culture, and streak it onto the agar plate. We're going to do this in a very particular pattern that I will show you now. This is how to streak bacteria onto an agar plate using the T-streak method. In the T-streak method, you will divide the plate into 3 sections, and you can, if you want, draw the T shape that makes those sections onto your plate, or you can just imagine them in your mind. In this procedure, you'll start by flaming your loop. And every time we flame our loop, I'm going to draw a little flame so we can just keep track. We flame the loop, and then you're going to use that loop to retrieve bacteria from the broth. And then you're going to streak the loop into the first section on the agar plate. That's going to be this larger section going across the top. And you're going to hold your loop sort of like a paintbrush and use soft gliding motions as if you were painting the bacteria onto the plate. So we start up here and just go back and forth, until that section has been filled. Then you're going to flame your loop again. And it's really important that you flame your loop in between these steps, because now you're removing that more concentrated amount of bacteria from the loop, and you're starting with a fresh loop before you start streaking into the next section. So in step 5, I'm going to streak the loop into the second section. So I'm going to use red just so you can see how it crisscrosses over. So you'll take your loop, it's now nice and clean, no bacteria on it. So you're going to pick bacteria up from this previous section by crossing into it once or twice, and then you'll do the rest of the streak in this section of the plate. Then after you're done with that streak, you flame your loop again. And now we're going to do the streak for the last section here. I'll use the green so you can see the overlap. And Same thing, we're gonna have a nice clean loop because you just flamed it, and we're gonna pick bacteria up from this second section. Just like that. And then this loop still has bacteria on it, so we need to flame the loop one more time just to clean it and sterilize it. The idea behind streaking the bacteria out this way is that somewhere here, depending on how dense the culture was when you started, somewhere here along these paths where you're dragging these bacteria through and kind of diluting them each time, you should be diluting down to the point that you are depositing single bacteria spaced out on the plate. And so up here, maybe you have a lot of bacteria that are all squished together. So when these grow, you'll just see a whole lot of growth all together. But then as you move through the sections, maybe by the time you get to this third section, you actually have some single bacteria that get deposited on the plate. And those single bacteria begin to use the nutrients in the agar plate and grow and divide. So that one single cell divides and divides and divides and divides until it forms a colony with so many bacteria in it that it's visible to the human eye. And so a colony is growth on a plate that is the result of one bacterium dividing many times. Each of these now is one colony. Now let's see it in person. I have my Bunsen burner lit and ready to go. I have my culture tube and I have my plate. I'm going to start by drawing the letter T on the back side of the plate. This is the side that has the agar in it, not the side that is the lid. I'll also write the bacteria that I'm working with and my own initials and the date. Now I'm ready to begin my tea streak. When you first pick up your bacteria, it can be good to give it a little flick to resuspend the cells in the liquid. Just make sure when you're doing this, you're holding it on the glass, not on the lid. The lids are not necessarily on snugly. All right, and then we'll use our aseptic technique. Flame the loop. Pull the lid off. Flame the top of the tube. And we're going to wait 30 seconds with the loop inside of the tube to let the loop cool down before we grab a loop full of cells. When it's cool, you can go in and grab a loop, flame the top of the tube, put the cap back on, and put the tube down in the rack. Now you can pick up your plate and begin the streak. And place the loop in the first section and just glide back and forth in that section. If you hold your loop nice and flat as opposed to up like this, the nice flat positioning will allow your loop to glide across the surface. If you hold it too upright, then it will dig down into the agar. So just a nice flat glide across the surface in that first section. Then put your plate down and flame the loop. Now that your loop is very hot again, we're going to wait 30 seconds in between the plate and the lid. I have the loop kind of sandwiched in between here so that the loop can cool without any spores in the environment from falling onto the loop and contaminating it. And then once 30 seconds is up, turn the plate over and you're ready to do your second streak. If you hold your plate at just the right angle, you can see the previous streak and make sure that you're crossing into it once or twice, and then keep your streak in just the second section that you're streaking in. Put the plate down, flame the loop one more time. We'll do another 30-second wait. And then turn your plate over. Again, hold it at the angle so you can see the previous streak. Pass the loop once, twice, and then stay in just the third section to finish your streak. Replace the plate, and one final flame of the inoculation loop to clean it. Now your tea streak is complete. It's ready to go in the incubator and see if you get any single colonies growing.